Hi everyone, my name is Maiko and I'm a grad student scientist at UC Berkeley. I'm here to tell you today about my work using frogs. But before we get to the frogs, I hope you'll take a little journey with me. Let's imagine that we're outside. Take a look around you. What do you see? Trees, birds, maybe there's a flower bed nearby. Or maybe you notice the ants crawling on the sidewalk next to you. Regardless, there are so many different species and so many different types of organisms outside. It's really incredible, really beautiful, and really immense. Have you ever thought about how evolution creates a new organism? How do you build a new species? But also, how do you keep all of these species distinct and separate from each other? There must be an evolutionary mechanisms to make sure that each species stays separate and that they don't randomly start to hybridize and mix with each other. Sometimes this hybridization helps in the evolutionary process when two different species come together to form a brand new organism. But this doesn't always work. Building an organism also is very challenging. Cells and DNA have to go through very coordinated movements and various different other processes in order to make this happen. One of the very first things that happens in early development is cell division. And this is where my research comes in. Our lab uses frogs to study the process of cell division and how different species stay distinct from each other. I use two closely related frog species and try to create a new organism by hybridizing them, by mixing both genomes and trying to make an embryo that has a mixture. However, this doesn't actually work very well because even though this new embryo can go through the first early stages of development and the first few cell divisions, during this process they're running into a really big issue. Normally during cell division, your DNA gets replicated and then separated faithfully and equally into two resulting cells. Both cells must have the same DNA information or else a whole host of other issues will arise when DNA is gained or lost. This is actually commonly seen in cancer, but this is especially crucial during early development. My work focuses on a really specific part of the genome, known as the centromere, which is the center part of your rod-shaped chromosomes. Now, the centromere is actually an incredible region of your DNA because it is absolutely essential in the process of cell division, and the DNA sequence as well as the proteins surrounding the centromere are fast evolving. So first of all, the centromere is necessary for all cell division because without it, you can easily gain or lose bits and pieces of DNA aberrantly and incorrectly. This is really bad for the organism. But paradoxically, the centromere evolves really quickly. So different species have different centromeres, even if they're really closely related. In the example with my frogs, if we take two species and try to make a new embryo, their centromeres actually realize that there's a huge mismatch between the maternal and the paternal centromeric DNA as well as centromeric proteins. Because of this mismatch, the cell actually cannot assemble the centromere very well on specific chromosomes. So in these embryos that I generate, they lose some paternal chromosomes, but with very high specificity. Because of this chromosomal loss actually leads to a very tragic metabolic crisis in the embryo, and so the embryo is actually not viable. However, we use this system to shed a lot of interesting evolutionary light onto the centromere. This locus in the genome is so important, is on everyone's chromosomes, and across organisms has the exact same function. And yet, different species have very different centromeres, making them incompatible with even closely related species. This provides a really interesting potential mechanism that evolution can use to make sure that different species stay distinct and don't try to create new organisms on their own. Now, I hope that I've told you that our frog system is a great way to accomplish this study, but also that you can learn to appreciate the diversity of organisms around you even more. The next time you're outside and think about the various species and organisms that you're seeing, maybe consider their centromeres. How different are they? Are they similar or potentially extremely divergent from each other? It's incredible to think about that diversity at the genomic level as well as at the organismal level, and brings a new appreciation for the biodiversity that we have all around us. Thank you.